I've tried to endeavor in my career to work with people way smarter than I am with the hopes that it rubs off. And so I would like to acknowledge my co-authors here. In this case, that was definitely the case. So I want to talk to you a little bit about the Wood Buffalo sponsored science that's been going on for the last 8 or 9 years, looking at source apportionment to understand the sources and the deposition of pollutants in the area. And that involved both an atmospheric and a terrestrial endpoints. So the ambient part, there was 2 studies done in Fort McKay, one in 2010 to 2011, which I'm going to talk about today, and one that was done in 2014 to 2015 that focused primarily on PAH species. There were 2 lichen collection studies that were done, one in 2008, which we've previously published, and the one that I want to be mainly talking about today, which was done in 2014. Also, as part of this program, we went out and characterized sources. And so looking at bulk materials to represent fugitive dust, DRI was hired to come in and do stack tests of upgraders and heavy haulers. And of course, we collected samples to try to evaluate the impact of wildland fire. So as I mentioned today, I'm going to talk mostly about the 2014 lichen collection. And I want to touch back a little bit with the 2010-2011 ambient work at Fort McKay. So the goals of the source apportionment are to elucidate the relationships between and spatial distributions between PAHs and epiphytic lichens. In this case, we use Hypogymnia physoides, which we did a work on and determined that to be the best indicator species in the oil sands region. And we wanted to calculate and quantitatively apportion the sources of PAHs. So you've heard a little bit about source apportionment today already, right? And that's trying to link sources and receptors. And there's 2 different ways of doing that. One is through deterministic models, where you start with the sources and all the understanding that you have of that source, how much it's emitting, what the chemistry is involved, what meteorology is there, and deposition parameterizations, and then you can estimate the impact of those sources to the receptor. The opposite version of that is a receptor-based approach, in which you make comprehensive measurements at a receptor location and statistically model back to the source. And I view both of those approaches as being independent and complementary of one another. So why lichens? Epiphytic lichens are ubiquitous in the oil sands region, and they're SO2 tolerant. And they're effective accumulators of PAHs, trace metals, and sulfur and nitrogen. And they provide a cost-effective way to put out a high-density network in an area where logistics prohibit us from taking active samples. They receive all their nutrients from atmospheric deposition, and the uptake occurs over the whole body, and they're not restricted by biological control. So source apportionment, I don't visualize as being just a modeling activity, right? If you look at it holistically, it's picking out the right sampling methods, matching it with the correct analytical methods, match with the correct model, and then going back and evaluating them to make sure they make physical sense. So really quickly, I want to step through the sampling. We went through, and Keith Puckett, the team's lichenologist, went out and collected a large volume lichen sample from multiple trees at each site. For the 2014 study, we used 127 different sites, picked from 2 separate host tree species, primarily jack pine and black spruce. Some of the sites we collected off of both. And just one comment, the spatial coverage on the northern area was just limited by the absence of the host trees. So those samples were all brought back to the lab and meticulously cleaned in a clean environment to pull out the bark and all the other non-lichen elements there. And then we sort of split them off 2 ways. So the samples that we were going to analyze for trace elements were sent to the University of Michigan, where we used zirconium ball mill to mill those down and homogenize them. And the organic samples were sent to RTI, where we did freeze-dried milling in a stainless steel ball mill. Real quickly, in terms of analytical, the organic samples were sent again to RTI, where they extracted them, they cleaned them up, and they analyzed them both with GC mass spec and some time-of-flight instruments for the alkylated pHs. And they employed a method to get all 16 of the EPA method, TO13 PAHs, plus a couple of the other species that are listed here. The inorganic samples are analyzed for total sulfur and nitrogen by Pacific Soils using dry combustion. Then what we tried to do was a total extraction of the lichens, where we broke down both the biological and the mineral matrix and analyzed them with a dynamic reaction cell ICP. And then a subsample of those we took back and analyzed via high resolution magnetic sector ICP. So in terms of the modeling, usually when you're doing source apportionment modeling of air quality data, your data is from a particular site, so you're spatially fixed and temporally resolved. Trying to do source apportionment for lichens is kind of the opposite, right? Now you're temporally fixed and spatially resolved. And so we plotted the data up just to make sure we had adequate variance in the data set. And so what this plot shows is we did not use AR6. In this case, I plotted the sum of PAH concentration as a function of distance to the closest upgrading facility, mining and upgrading facility. And similar to the speakers before me, who I'd like to thank for setting me up perfectly for this talk, is, previously we were looking at that 40 to 50 kilometer range, and when we started mapping them to the closest source, that, as the other speaker says, that tightens us up into the more 20 kilometer range. And what no one has said is this is textbook. This is absolutely textbook of what you would expect for dry deposition of coarse or supermicron particles. This is just an example of some of the individual PAH species. I picked two of the higher molecular weight species, so chrysene, which is a 4-ring compound, and benzoate, which is a 5-ring compound. We see this absolute perfect pattern carried through, and of course there's a lot of autocorrelation between the different PAH species. You look at the second plot, and that just shows you our interpolated plot in terms of the area of impact in the sampling domain, which is about 150 kilometers, centered around Fort McKay. So we plotted up also some of the trace metals, and for the most part, these looked exactly like the results we got from 2008, with the exception of those 3 outliers that I sort of have circled there. And when we plot them up, those are areas to the south near Conklin and maybe a little bit north, which did not appear in the 2008 data set. So we zoom in and we can see those sites. 3 of those sites were what I would term kind of near road, but also looking at the satellites, we see there are areas of disturbance down there that could lead to some of those trace element enhancements that we saw. So we used the positive matrix factorization model and fed in all of this lichen data. We got a very robust 7-source numerical solution. And so within the model, there are different ways that you can characterize the goodness of fit and how numerically robust it is. And both the displacement and the bootstrap results were actually quite good, so we're very happy with this model. I don't expect you to be able to read these species at the bottom, but basically, the modeler's looking at this output. Both the presence and the absence of known tracer species allows us to quantify what those sources are. And so in this plot, not only do you see what we think the sources are, you can also see when we take those source contribution estimates for each sample, then we can interpolate that spatially as well, which is the maps that you see here. And underneath, when you look at the quantitative apportionment, we can see how much of the sum of PAHs was explained by that particular source. And so in terms of the sum of PAH, this fugitive oil sand factor is what we believe is really driving what we see. And as you can see, we can see 52% of the total deposition is controlled by that one factor. The other 2 fugitive dust factors on here have non-significant contributions to PAHs. The other 2 that were significant were the 2 combustion factors, the first of which we sort of attributed to heavy-duty hauler or heavy-duty vehicle transportation, which explained about 12% of the sum PAHs. And then the secondary combustion, which we think is a commingled source between some stack emissions and wildland fire emissions. That explained about 21% of the PAHs that we saw. So when you looked at the entire study domain broken out by each of those PMF identified sources, we can see that fugitive dust contributes 58% of sum of PAHs in the entire sampling domain. If we sort of narrow our focus down to just that 25 kilometers range around all of the major production facilities, then that fugitive dust factor increases up to 78% of total PAHs being able to be explained by those. So the conclusions of the Lycan study, as I mentioned, is that these fugitive emissions, primarily from oil sand operations, dominate the spatial distribution of those higher molecular weight PAHs. The near-field lichens also had significantly higher concentrations of those petrogenic PAH species. And again, the use of all the major source locations rather than that midpoint site shows us that sphere of influence shrinks from 50 kilometers roughly down to like 20. And again, very indicative of coarse particle dry deposition. So we want to go back and revisit to say, does the results of the PMF model make physical sense? And to kind of do that, we're going to ask ourselves that question. Is it reasonable that fugitive dust is the major contributor to atmospheric near-field deposition of PAHs? And to do that, I want to link back to some of the ambient work that we did at Fort McKay. And again, this is the 2010-2011 study. The previous speakers have identified where the Fort McKay AMS-1 site is located. And as you can see, it's surrounded by lots of different production facilities. During this time period, we collected daily sequential dichotomous samples. So we had both fine fraction and coarse fraction sample pairs. Out of those 392 pairs, we selected 100. We had a little bit of a budgetary constraint on the chemical analysis, so we selected that subset. And of course, during that time period, we were impacted by the 2011 Richardson wildland fire. And so for the PM 2.5 source apportionment results from Fort McKay, we had a 5-source solution. The first of which was a fine fraction component of fugitive dust. And the one thing that I just wanted to highlight is that what we found when we looked at that temporally resolved data is that the winter time, when the ground is frozen and there's snow on the ground, we saw very little, if any, impact of the fugitive dust that we were seeing for the remainder of the year. We also saw the impact of the Richardson wildfire. These are the samples that were valid. So we had like 7 samples that were invalid because when these samplers collect so much material and it restricts the flow rate of the instrument, it shuts itself off. And so we had a large number of samples that were impacted by that phenomenon. The other interesting thing is that during the winter, we were seeing impacts from biomass burning. So whether that's residential wood heating for residents or clearing burning activities of the operators, it's unknown, but we clearly see an impact during the winter of biomass combustion. And some of the other sources that you see here, we also see salt from road de-icing in the wintertime only. And then we see this oil combustion factor all year long. In the case of the coarse fraction, we have a robust 6-source solution here, of which 3 of them are fugitive dust. And we see this biomass combustion factor. In this case, we see the Richardson fire. And again, the 2 main fugitive dust factors are either extremely small or non-existent during this time period. So the implication there is if you go back and you're sampling snow and you're trying to get an idea of what fugitive dust deposition would be, you have to question how representative that would be to the rest of the year, for sure. And so when we look at our quantitative source apportionment of the PM at Fort McKay, here's our fugitive dust. For the PM 2.5, we get about a quarter of that mass is explained by fugitive dust factors. And from the coarse fraction, we're seeing northwards of 80% being contributed by those 3 fugitive dust sources. So that when we talk about does the lichen results seem reasonable, based on what we know about the ambient particulate matter in that region, I think it is reasonable. The other thing I just wanted to mention is that when you look at the overall concentration of PM 2.5, and one thing I didn't mention is there's 2 columns here. One includes all of the data, and then the other column that I'm kind of highlighting is with those 2 exceptional wildland fire events samples removed. But on average, the PM 2.5 mass is actually relatively low there at 5.6 micrograms per cubic meter. If we were to include the impact of the 2011 Richardson wildfire, that would actually increase the annual average concentration up over 20 micrograms per cubic meter. Wildland fire has a big impact on PM at the site. In terms of the coarse fraction, and this is also kind of interesting, it's higher, the concentrations in the coarse fractions are higher than the fine fraction, which is sort of an unusual finding unless you're in that area of anthropogenic source influence. And so in terms of the future work, we've analyzed a lot of these samples for the C1 and C2 alkyl PAHs. We haven't had a chance to actually incorporate that into our analysis yet. Likewise, we've analyzed all these samples for stable lead isotopes, and we have not put those into the analysis yet. And then at some point, we wanted to integrate the ambient work and the lichen work in a more formal way, and then go back and look at our temporal analysis, because there were lichen collections done in 2002, 2008, 2011, and 2014. So I see by the clock I have a little bit of time, so I have some bonus material to go through with you. So here's a teaser for the temporal resolution issue. So I went back and looked at this fugitive oil sand factor that we saw both in the 2008 and the 2014 analysis. And what you can see is that spatial distribution sort of getting a little bit larger, potentially as some of these new production facilities go online. If we look at the tailing sand factor from the PMF model, we see a little bit of oscillation, and again, we pick up some of those activities now that are going on in the southern area of our domain. And I want to talk a little bit about Fort McKay. And so the interesting thing when we look at those results, this is a windrose from that 2010, 2011 time period, is we really do have this bimodal wind direction feature there. And I think that's a feature of being in that river valley, and especially during the wintertime, you get these subsidence events, and the air just settles in that river valley and swashes back and forth. And so I built a three-dimensional map on which to plot, in this case, benzoapyrene, using that interpolated data from the 2014 lichen collection. And it really shows how the impact really is sort of limited and funneled down by that river valley. So it's not only where the sources are located, it's what's the meteorology and what's the topography of the area that's driving those impacts. So if you're looking at water impacts, potentially that's of interest, where it is in the river valley. And with that, I'd like to thank you for your attention and be happy to answer any questions if there are any.
Ian Marina, Associates. Just a question, where's the bitumen and the oil sand ponds themselves? Where does that show up as a source in your data?
So in the 2008 work, we actually separated things together in terms of what we call process materials, and we have the oil sands. And so chemically, when you're looking at the data and at the profiles, that oil sand factor has both the petrogenic and the crustal elements in there. And then once they're processed, that tailing sand end member goes one way and the bitumen goes the other. And we just didn't pick up, either in the lichens or in the most case for the ambient side, that what I call process material, that end member that has the petrogenic endpoints in the absence of the geological material endpoints. And so, I know a lot of people have highlighted this issue of petroleum coke, right? And that would put you down on the petrogenic side. I didn't mention the lead isotope work, but interestingly, you've got these two end members, right? You've got the geological material end member in stable isotopes, and then you have this petrogenic end member. And almost in the lichens, we see everything ranging from that middle mixed member, which is the oil sand, and then leading up to the right, which would be just your sand in the absence of the petrogenic component. We did not see anything at all in that lower left-hand region, which would be indicative of that bitumen or petroleum coke impact. So we're just not seeing a whole lot of that from either the lichen measurements or the air measurements at Fort McKay. Does that answer the question? I can't remember where in the room that came from.
Bill Shoddock, University of Alberta. Matt, great presentation. Have to ask the question, how old are the lichen samples?
So this is the question. So the lichenologists that are on our team and other people we've asked, I guess the question you're really asking is, when we take that sample, what's the temporal frame of reference of what that represents, right? And when we originally started, they were guessing that growth would potentially be 2 to 3 years. And so if they're doing a lichen collection every 4 to 5 years, we should be able to see the needle moving. as a homogenized group of both younger individuals growing and some of the older individuals growing. That's the best answer I can give you.